So pretty much all of class today we're going to be talking about one main component in sort of genome surveillance and guardians of cells and life and death decision making, and that's the tumor suppressor P53. Okay, so if you had to, we have lots and lots of tumor suppressor genes, by the way, at least a dozen. But P53 is the most important one because it's the only one that can function in the most cells at most parts of the cell cycle and can elicit all the physiological outcomes that you'd want to have in response to DNA damage. Okay? So again, DNA damage is sensed by a family of stress-activated protein kinases that are called ATM or ATR. They each recognize different types of DNA damage, but what DNA damage, if you think about it, what it always has in common are exposed regions of single-stranded DNA. In fact, the easiest way to trigger a DNA damage checkpoint is to just take some single-stranded DNA and chuck it into a cell, and it works great. Okay? Remember, our DNA is generally double-stranded all of the time. The only time that you have exposed regions of single-stranded DNA, for example, is during DNA replication. Obviously, when you have to separate the two strands and generate new daughter strands. But that occurs very quickly. Remember, DNA replication is fast. It goes at about 500 nucleotides a second. So you're not going to have exposed regions of single-stranded DNA very long. Transcription is the other time, because you have to separate the two strands of DNA to transcribe the template strand into messenger RNA. That also goes pretty fast, about 50 nucleotides a second. So your cells normally do not ever have exposed regions of single-stranded DNA. That's why it's a good signal to activate these particular kinases. Okay? So the key thing that you want to do in response to perceiving single-stranded DNA, first and foremost, is stop the cell cycle. You don't want the cell to just keep on going, because if it's going to go into DNA replication, you don't want that to take place until you've had an opportunity to fix the damage. If you're already in S phase or after S phase, you don't want to finish it, and you sure don't want to go into mitosis with DNA damage. So we've got these cell cycle DNA damage checkpoints that can stop the cell cycle at any point when there's damage. We have a P53 dependent checkpoint, and we have a P53 independent checkpoint. And generally, if you take a look at the slide, you can see that you get both of them activated at most places throughout the cell cycle, with one glaring exception, and that's up here during G0 to G1, which is the critical phase, because A, that's where most cells are at any given time, and B, it's damage that's going to take place that you want to fix before the cells start entering DNA replication. And that is only the P53 DNA damage checkpoint. Okay? The two checkpoints work differently. We'll get into that in just a second. But the big picture is this. P53, as you're going to see, is a DNA binding transcription factor. And it works by activating or repressing the expression of target genes. The gene that's required for P53 to activate the DNA damage checkpoint is a little protein called P21CYP or P21KIP for CDK inhibitory protein. P21CYP is great. You express it and it will bind to any cyclin CDK combo. Doesn't matter if it's an A, a B, a D, an E cyclin, or a CDK 1, 2, 4, or 6, shuts them all down. Stops the cell cycle right there. Okay? So that's how P53 works. The other one are these other two kinases, check1 or check2, that are also activated by the ATM or ATR kinases. And they go about it differently. And that is the P53 independent checkpoint is that they are also going to inhibit cyclin CDKs, but they do it by inhibiting CDC25. So Basically what they're doing is that they're inhibiting the phosphatase that would otherwise activate cyclin CDKs by dephosphorylating threonine 14 and tyrosine 15, which is put on by WEE1. So guess who's the most important component in the p53 independent checkpoint? WEE1. The cell is using the same mechanism to stop the cell cycle due to DNA damage here by inhibiting CDC25. It's using the same mechanism that it already has to stop cells from going into mitosis until they've doubled in size. So it's not reinventing something new here. It's just reutilizing or employing a mechanism that it already has to inactivate cyclin CDKs normally during the cell cycle. So it's not using a brand new mechanism to do that. Okay, so here's what's going on here. The ATM or ATR kinases, let's go through the p53 dependent pathway first, okay? Here's what's normally going on in our cells. p53 levels are typically found in all of your cells, so you'll find p53 in every cell all of the time at a vanishingly small level. It's a constitutively expressed gene, and there's a vanishingly small amount of p53 protein. The reason is it's very unstable. And the reason it's unstable are twofold. First, it has to be phosphorylated to be stable. Secondly, it has to bind DNA to be stable. So DNA actually is an allosteric effector of p53 because it stabilizes it by binding. DNA binding requires that p53 become a tetramer. It only forms a tetramer efficiently if its level accumulates enough to come together and form these tetramers. That requires phosphorylation. So it's all interconnected. So what we have to do, though, to start stabilizing p53 are two things. First thing we do, let's just phosphorylate it. That starts it on the path to accumulating. So think of what's happening now. We phosphorylate p53, its level is going to start coming up. It's gradual. It's slow. The other thing that ATM kinase does, it phosphorylates a protein over here called MDM2. The reason p53 is so darned unstable because MDM2 is a p53 specific ubiquitin ligase. Our cells have a ubiquitin ligase that recognizes p53 and nobody else. And its job is to keep the levels of p53 very low in your cells. Okay? So, we can stabilize p53 by phosphorylating it, but what we want to do is get rid of MDM2. And there's two things that are going to happen here to do that. The first is MDM2 gets phosphorylated by the ATM kinase. So it gets phosphorylated at the same time p53 does. That inactivates its ubiquitin ligase activity. Okay? So now we're also going to help p53 become more stable. As if that's not enough, we're also going to take MDM2 and sock it away in a different compartment. All of this action that you're seeing here is in the nucleus. What we can do is we can take MDM2 and bang it out of the nucleus and put it in the nucleolus. Remember, the nucleolus is a little subcompartment within the nucleus where ribosomal RNA and ribosomal RNA synthesis and ribosome assembly take place. There's a little protein there called ARF. ARF will bind MDM2 and take it out of the way. So we're taking out this p53 ubiquitin ligase two ways. One is we phosphorylate it. The second is we shove it into a different compartment. So now your p53 levels will start to go up. As you're going to see, p53 binds to DNA sequences associated with a large number of genes. There is a consensus p53 binding site. The consensus just means just that. It's a consensus. They're not identical. So what it means is p53 has high affinity DNA binding sites, medium affinity, and low affinity. and it's going to occupy those different DNA binding sites based on its concentration. At its lowest concentration, as soon as we start stabilizing it, its highest affinity binding site is found on the p21 CYP gene. And so the first thing, as soon as we start raising p53 levels, is p53 is going to bind to the p21 CYP gene and activate it. So now we make p21 CYP. It's going to bind to whatever cyclin CDK is there at the cell. Doesn't matter what part of the cell cycle you're at, and stop the cell cycle. Okay, now we've stopped the cell cycle. We've also got a backup way to do that, so long as we're not in G0 or G1. So the same time that we got this action over here going on, we've got this kinase cascade that's going on over here. And the kinase cascade is an ATM or ATR, activates another one or pair of serine-threonine kinases called CHEK1 or CHEK2 for checkpoint kinase. Now we got some redundancy here, because CHEK1, CHEK2 can also phosphorylate p53. They can also help make it more stable. So we've got some redundancy here in phosphorylating p53 either by ATM or CHEK2. But ATM is the major driver for doing this. Okay? The main function of CHEK1 or CHEK2, and again, you can put a 1 or a 2 here, it doesn't matter. They phosphorylate CDC25. And when you phosphorylate CDC25, that causes it to become ubiquitinated, and it gets degraded. Okay? Well, now that what that is, that's another mechanism to stop cyclin CDK activities independently of p21 CYP. Because if you take out CDC25, that means that the CDK subunits that are phosphorylated by WEE1 are going to remain phosphorylated. Okay? So you're using WEE1 as the decider here. When you take out CDC25, that's what's also causing the cell cycle to arrest. Okay, so now you've got the cell cycle arrested. Two different ways of doing it. Okay? Now you're not going to go any further, but you still got this damage. So this pathway remains active. We remain in this cell cycle arrest, and p53 keeps going higher and higher. Now we get to the next concentration level. Okay? And what's going to happen here is, just trip over to the next slide. It's a pendulum of what's going on here. So now what's happening is, p53 levels, it's basically going increasing. Now we're moving somewhere over here, and you can see that what we're doing is we're going to start tipping the balance of what the cell can do. Now we're going up to an intermediate level of p53. It's now going to bind to lower affinity targets, but they're not the lowest. The next targets in line are the enzymes that can fix the damage. Makes sense. This is why you stop the cell cycle, is because you want to be able to repair the damage. We have all these different cohorts of DNA repair enzymes that are normally not transcribed. p53 kicks them into action. So depending on the type of damage you have, you will activate the appropriate type of repair enzyme that can go fix it. Okay? Now let's assume the damage is fixed. Okay? So let's say you succeeded. You go through, you activate those DNA repair enzymes. Well, now we no longer have DNA damage. The target is gone. That now means the ATM or ATR kinases, they're shut off. Okay? Well, now the phosphatases that are always there all of the time reverse the phosphorylation. So now that means MDM2 becomes active again. MDM2 will also now bind to p53, which is also dephosphorylated, and now our p53 levels drop very, very rapidly. Okay? So now what happens is p53 levels drop to where they can't sustain p21 CYP expression. We turn that gene off, and after a little while, the cell just picks up and goes through the rest of the cell cycle like nothing happened. Ideally, that's what you want to take place. And the vast majority of the time, that is what takes place. The system works. You stop the cell cycle, fix the damage is gone, p53 levels drop, we get back to where we were going. Okay. What happens if you don't fix the damage? That stays active. This continues to rise. So now you're going to see why our cells, why they're so concerned on keeping p53 levels so vanishingly small. Because what happens is, if our p53 levels keep crossing up to this point, we're going to go over a threshold. And once we cross that threshold now, that only occurs at very high levels of p53, the cell is going to execute its death program. It's going to undergo a process called apoptosis. It's going to commit suicide, and now you solve the problem in a very, very terminal way. That cell that could not repair its damage, goodbye. It's gone. Okay. So let's stop for a second here and take a look at apoptosis. Okay? Apoptosis is also perfectly normal. Okay? And what I mean by that is, hold up your hands, look between your fingers, and you realize you don't see anything there. You used to have webs there. Okay, so back in our fetal days, when life was good and comfy, we had webbed digits between our fingers and between our toes. They're gone now. Why? Those cells underwent apoptosis during fetal development. They're gone. Any place in your body where you've got an opening, an orifice, or a cavity, cells used to be there, and they were eliminated by normal apoptosis. So the normal fate of a lot of cells during normal development is they execute that cell death program called apoptosis. It's called programmed cell death, or PCD. Considered the ultimate in a terminal differentiated state. Okay? And in those cells, they can elevate p53 levels normally. They have a different mechanism of doing it without undergoing DNA damage. The consequence of a failure to activate that program is you can wind up with additional cells. You can wind up with webbed fingers or feet like a duck. And that's all a consequence of the failure to execute apoptosis. So apoptosis, or death, is a normal physiological process. And as Lewis Thomas noted in one of his essays, it's ironic that death is in fact a normal biological process. We have a genetic program that's already in our cells that can be executed under particular conditions that cause normal cells to die. Okay? Our mortality is built into our cells. Now, what we're doing to activate apoptosis in the case of cancer is to basically call upon that mechanism to go to work under extraordinary circumstances where we've suffered damage to our DNA. Okay? So let's take a look at this death program and what it entails. Okay. And we'll actually take a look at some videos because it looks cooler to watch than it does to just kind of look at it. Apoptotic cells, as you're going to see, undergo some bizarre conniptions. Because what it is going to require, what lies at the heart of apoptosis, is activating a battery of very specific proteolytic enzymes that are called caspases. And what caspases do is that they cleave inactive precursor proteins that are going to be around all of the time, and we'll get into those in a minute. And what it does, it converts them to where they are now active. And what these active proteins do when they're activated by caspases is, for example, as you'll see, they start cleaving your cytoskeleton. The cell's going to collapse. Your membranes fall apart because there's no cytoskeletal structure to hold them in place. Your nuclear membrane disappears. Your chromosomes get chopped into little tiny pieces. This is one of the clearest signatures of a cell that's undergoing apoptosis is that if you take a look at the DNA, if you isolated DNA from your cells, it's very, very high molecular weight. You run it on an agarose gel, stained with ethidium bromide, you see a band up here. It's very large. It doesn't migrate far into the gel. It's very, very high molecular weight. Take a look, though, if you start isolating a cell, DNA from cells that are undergoing apoptosis. Over time, what you start to see is the appearance of a ladder. And what's happening is your DNA is being digested by an enzyme called DFF40, for DNA fragmentation factor 40. That's an enzyme that's normally inactive until it gets switched on by a caspase. And what DFF40 does is it cleaves your DNA in between adjacent nucleosomes. So it starts chopping your DNA into little pieces in between nucleosomes. Now remember that the distance here of DNA in a nucleosome is about 200 base pairs. So what you start seeing is a ladder to where your DNA is being broken down into little nucleosome-sized bits. And these are multiples of 200 nucleotides. So that's 200, 400, 600, 800, and so on. Because you start chopping this, and then you ultimately break them down into 200 base pair bits. Once you've reached that point, it's irreversible. You ain't coming back. Because think about it. You can't take 3 billion base pairs of DNA, chop it into 200 base pair pieces, and say, put this back together again. It's like Humpty Dumpty. Once you're fragmented that far, lights out. So what happens is, your DNA gets chopped into little pieces, your cytoskeleton's chopped into pieces, the cell starts blebbing, breaking up, and fragmenting. As you're going to see, it literally explodes. And what you're left with is a little thing called an apoptotic body, which is a little inert clump of crud that's going to be engulfed by a macrophage, and it's gone. You never knew the cell was even there. It doesn't leave any remnants. Okay? So that's apoptosis. All right? Now, it's normal, but we're also looking at it under situations where we can induce DNA damage. Okay, so what we're going to do is take a look at a couple of time-lapse videos that actually show this process occurring under a couple of different circumstances. And then we're going to look at a computer graphic animation. It's not as cool as the first one that we saw. And this actually has a soundtrack. But I don't think cells really make this noise when they're undergoing apoptosis. And then we'll look at the mechanism. Okay? All right, so let me call up, let's see here. Here's a video. Let's watch this one first. Okay. This is a cell that's going to acquire another nucleus. Okay? This is a cell that we're artificially going to shove in a nucleus, okay, that is undergoing DNA replication. This cell has no clue what's coming. Okay? And what's going to happen is all of a sudden this cell, which is somewhere in the cell cycle, is now confronted with a new nucleus that's in the midst of DNA replication. It's going to have a lot of single-stranded DNA exposed. But this cell is going to go, WTF, what's going on? And you're going to see what goes on here. Okay? This also, you guys, this gives you a sense of the time frame. Don't look at my airplane. Okay, here we go. All right, there's the ending, sorry, spoiler alert. Okay, here we go. Here comes the nucleus. And then this is what you're left with, this little tiny clump that normally a macrophage is going to come through and take it out, engulf it. Here comes, yeah, I mean, that's pretty spectacular. It's just splat. It's like a paintball. One more time. Here it comes, boink. Doom. Okay. Soundtrack option. Okay. There's one way you can cause apoptosis. That sort of shows you single-stranded DNA. Now here's another harsher way. m4 cell that you're going to see is being driven to proliferate by having a MYC oncogene. Okay, so it's overexpressing MYC protein, which remember is all the way down there at the bottom of the RTK signaling pathway. Okay? MYC, remember, is responsible for activating cyclin D, so the cell wants to proliferate. So this is a cell that's primed to remain proliferative. And what you're going to do to this cell, not you, but what whoever nicely made this little video did, they're starving it. So you've got a cell that's being driven to proliferate by MYC oncogene. And what you're going to do is grow it in medium that lacks glucose. Nice. So the cell is being starved at the very time it's being told, go for it, proliferate. And what you're going to see is the cell can start trying to do this for about one round of cell division. And then it's going to get very confused, and you're going to see this cell on a desperate hunt looking for glucose. And then you're going to watch what happens when it doesn't find it. How to make an Oscar about this? Okay, here you go. This also, again, kind of shows you how cells are naturally sort of twitchy, kind of walking around. Oh, there we go. It undergoes division. Here's the daughter cell. I just divided. MYC is saying, let's do it again. There's no glucose. Looking, boom. Sad. And you can see that this took about a day. Okay? So it's a cell that basically went through one round of mitosis. It's got that MYC oncogene that's still driving it, so it's going to try it again. There you go. By 30 hours, there's the little pycnotic body. Okay, so let's just... It's not as dramatic as the first one, but it kind of gives you the idea of another way that you can force a cell into undergoing this process. You can see how it's shooting out all those pseudopods. You got a lot of microfilament action going there. And then that's it. So can you guys also see this doesn't happen immediately? It takes several hours to undergo. Okay, so what's going on here at the molecular level? So here's a computer graphic animation of it that was developed by some guys down in Australia. And so now what this is going to show you is a video depiction of apoptosis. Now this is apoptosis that's going to be triggered by the binding of a cytotoxic T cell, which is going to cause the target cell that it binds to activate a signaling pathway that will cause it to die. We're not concerned with the signaling steps that go on here. We're only concerned with the actual process of apoptosis that it's going to trigger. So the process that you're going to see is exactly what you're going to find in a tumor cell that's going to be overexpressing p53. Okay? So let's watch this. Good. We got everybody hear the sound? I might as well turn the lights out since it's going to be lights out for the cell here. Okay. Okay, here comes the cytotoxic T cell. This is Kittleson's favorite part of the movie. It's all downhill after this for him. Told you, here you go. Yeah, like you really think it sounds like this? It gets better. And that's actually what these things are called, death ligands and death receptors. basically binding adapters that are just like Grab2. Okay, here come the aliens. Now you see your mitochondria getting involved here. APAF1 is the activator of apoptosis. And it's going to assemble to make this little funky brain. And this is the apoptosome, which is basically a caspase factory. There you go. This was at the Melbourne, Australia. Drew Barry's real. Here we go. Your life of the cell. No, we're not going to show that again. We will, but not today. That's for your final exam. Okay, so did that give you kind of a graphic appreciation for what's going on inside these poor cells? Okay? Understand, this is normal. This is normal for certain cells to undergo this process. The only thing that was left out at the end were all those little fragments being engulfed by macrophage and cleared out. So they're gone without a trace. So what's going on here to explain everything that's occurring? Okay, this is a decluttered version of the pathway. It's a pathway that you guys can think of as a caspase cascade. It's going to involve P53, okay, we need it. It's going to involve the mitochondria now, okay? And this is one of the things that you're going to see that explains why cancer cells do not metabolize glucose by aerobic respiration. They want to stay the heck away from the mitochondria. And that's why cancer cells exhibit something called the Warburg effect, where they respire aerobically through glycolysis. It's a lousy way to make ATP, but it's a much more effective way of metabolizing glucose to make macromolecules that are required for purine pyrimidine biosynthesis and amino acids. It's more of a biosynthetically robust pathway, but it steers clear of the mitochondria, and you're going to see why that's important. Okay? All right. So we start off up here. Let's just take it step by step with what's going on and what P53 is doing. P53, as its levels are now way high, okay? So this is a cell that the DNA damage never got repaired. P53 is going to activate the transcription of the APAF1 gene. That's the inactive. This is the guy that's going to make the ring that's going to be the apoptosome. It's catalytically inactive, so please understand, I have to emphasize this, we're increasing levels of inactive precursors here. So we're going to make more APAF1, but it's not active. We're going to increase caspase 9, that's an initiator, and then caspases 3 and 7 are effectors. They're the guys that are really going to do the damage. So now the cell is kicking up the expression of the caspases. They're the little guys that are going to be catalytically activating all the other caspases. This is an auto-activating cascade. Meanwhile, P53 is repressing the expression of a gene called BCL2. BCL2 is a pro-survival gene. And BCL2 works by preventing apoptosis, normally. It's a survival gene. So P53, when it activates these genes by binding to DNA sequences, is also, when it binds to the BCL gene, blocks its expression. And that's going to be important, because what BCL does is that BCL is going to prevent the release of cytochrome C from the mitochondria into the cytosol. Cytochrome C is normally used for electron transport in the mitochondria. It stays in the mitochondria. It's the Las Vegas of your cells. What goes on there stays there. Shouldn't be out here. But now what we're going to get are two things starting to accumulate in the cytosol that are not normally there. Okay, here's what P53 is going to do. P53 is also activating the expression of an enzyme called ribonucleotide reductase. Ribonucleotide reductase does exactly what it says it does. It's going to take ribonucleotide triphosphates and reduces them to deoxynucleotide triphosphates. Now, why would you want P53 to do that when you've got DNA damage? Why would you want to increase deoxynucleotide triphosphates in the cell when you've got DNA damage? What are you going to use the deoxynucleotide triphosphates for? What are they normally put into? DNA. So normally, the cell is going to increase the pool of deoxynucleotides because the cell is going to use them to repair the damage. But we didn't fix the damage, remember? So what's going to happen if you don't fix the damage? P53 stays on, ribonucleotide reductase stays on, and now the cytosol is going to start filling up with deoxy-ATP among the other 3 nucleotides. Deoxy-ATP is an activator of APAF1. Because APAF1 is now sensing, hey, there's something out here in the cytosol that shouldn't be here. It uses it as an allosteric activator. P53 also increases the expression of an enzyme, of a protein called BAX1. BAX1 is a mitochondrial protein. It's a ginormous pore protein that basically opens up your mitochondria. They're normally sealed up. So what BAX1 does is that lets cytochrome C go barreling out of the mitochondria into the cytosol. And this is going to be our second activator of the APAF. So we've got 2 signals now that are being released into the cytosol. So now we got deoxy-ATP, that's coming from ribonucleotide reductase, P53 raised that. Cytochrome C is increased because BAX1 allows it to get out. So now the APAF1 becomes active. P53's been kicking up the levels of it, but now it's going to become active. APAF1 now converts these inactive Cas bases to their active form. Now they're autocatalytic. So all of these inactive precursors to the Cas bases activate themselves. Caspase 3, one of its targets, is active. It behaves just like cofilin. So caspase 3 goes over and starts chopping your actin microfilaments into pieces. That's why the cell collapsed. Caspase 3 also activates DFF1, or DFF40, sorry. That's what cleaves your DNA into little fragments. And so by that point, it's over, and the cell dies. Okay? So what you guys see here, it's a caspase cascade. There's no kinases involved here. It's all caspases. It's all proteolytic enzymes that are being activated, and the role of P53 is to crank up their levels. Can you see that none of this happens without P53? If P53 is not there, cells cannot undergo apoptosis. That means that if P53 is not there, here's where we need to start going back into looking at cancer progression. If you do not have P53, you can't block the cell cycle very effectively because you can't activate P21-CYP. You cannot fix DNA damage, okay? So, all of those carcinogens that you were exposed to, courtesy of Bruce Ames, guess what? You can't do anything about the damage now. You can't fix it. You can't stop the cell cycle. The cells are accumulating mutations. And now, they can't commit suicide. So now you've got cells that are going to keep rolling through the cell cycle. And as you do that now, cells that have acquired mutations that give them a proliferative advantage, you're selecting for them because they're going to keep proliferating. Okay. So what can happen to P53 that can take it out? Okay, here's where we need to focus on P53 itself. Here's the structure of P53. Okay? P53 has multiple functional domains. That's bad because it means there's a lot of things that can go wrong with it, just like the LDL receptor. So P53 binds to DNA as a tetramer. So here's what it looks like when the tetramers bind to DNA. Okay, there's the DNA strand. It kind of wraps around it. When it binds to those sequences, it's got a region down here that's required for transactivation. Transactivation means it recruits RNA polymerase to that gene and allows it to be transcribed. If we take a look at P53, let's go through and if we analyze tumors, okay? In this case, we analyze 26,597 tumors. And we look at the P53 genes in those tumors. What this pie chart is showing, for the cells that locked P53, they lost it. Almost 3/4 of them have missense mutations in P53. The missense mutations cause P53 to misfold and to be unstable. More unstable than it normally is. So the problem with these missense mutations is you go ahead and you go in, you try and activate DNA damage. The P53 levels can never rise to high enough levels to even activate P21-CYP. You can't stop the cell cycle, let alone activate DNA repair enzymes. Here's what's insidious, though. If you take a look at the P53 gene, if you take a look at where these mutations map, the majority of the mutations map right here in the middle part of the P53 gene. It's the region of the gene that encodes the amino acids that are required for it to bind to DNA. If P53 cannot bind to DNA, it can't be stable. Okay? What you find in the P53 gene is that these positions here are mutational hotspots. So it's not like every single nucleotide in the p53 gene is equally susceptible to mutations. So the notion that when you hear mutations occur at random, there's no such thing. Every nucleotide in a particular context has a different preference for being able to undergo a mutational event. And these mutational hotspots in the p53 gene, these all map to guanines that can be very readily changed by mutations that change them ultimately to a thymine. They result in missense mutations that cause p53 levels to never accumulate. So the problem with our p53 gene is, think about it. Here's probably one of, if not the most important gene in our genome as far as guarding our genomic integrity. We've only got 2 copies of it, one from mom, one from dad. If one of them undergoes a mutational event, you've now just dropped your ability to produce p53 by 50%. You've knocked the amount of p53 that you're going to be able to produce normally under conditions where you have to crank it up by half. You are only going to make half as much p53. That means your cells are going to be much, much more susceptible to not being able to stop the cell cycle or activate DNA repair. Okay? So that's the Achilles heel with the system. We put a lot of trust into one tumor suppressor gene, and as you guys can see, it's very easy to knock it out. Okay? All right, so on Friday, we'll pick it up and keep going through this and continue working on some other examples of campaigns. Okay? See you guys then.